Hello kids, this is the first podcast for the year. I'm just going to essentially try to highlight the troublesome areas and what you're going to need to know for tests in the final place. So first you have to know what the parts of a microscope are. This is simple. You're going to have to find a labeled item and say what it is and what its function is. And so I've labeled these items by function according to the stars on them. Go through them real quick. I did not label structural items, that includes the arm and the base, but I did label things that seems kind of intuitive, but their sole purpose is to position the sample underneath the lenses so that you can see them or see a specific region of your sample. Those are the stage that actually holds the sample, includes the clip, the coarse and fine adjustments. The coarse is on the inside and the fine is on the the coarse is closer to the arm of the microscope, the fine is further away from the arm. Those adjust the focus by moving the stage up and down. Note that you only use the coarse adjustment when you're on the lowest power, the scanning power, the 4x lens. That's the only one that has enough clearance that you don't risk smashing the slide against the objective lens, damaging the sample and the lens. And then there's knobs that control the X and Y coordinates on the stage. Those are all about moving the sample around. The main purpose of a microscope, as you know, is to apparently magnify the light so that you can interpret it. And so I've marked those with a purple star, and I left one of them tricky. But first, so there are two lenses that actually do the magnifying. The ocular lens is within the eyepiece. It is set at 10 times magnification. And there is a variable objective lens. It's called the objective lens because it's close to the object that you are investigating. Do not use the 100x lens in our Gen Bio laboratory course because it is an oil immersion lens. It will not work, and it might just make things messy. Now, technically this item down here, the condenser, does bend light, and so I couldn't help myself. I had to label it with that star. However, it is not important at all in magnifying the image for you. What it actually does is gathers the light from the light source down here and beams it back strongly, condensed, onto the sample. Because one of the objects of magnifying lenses is to actually reduce the amount of light that's going into the eyepiece. This is there to put more light back on it. So the other things that control that, the iris diaphragm is a slider just underneath the stage. That's mostly so that you can find a comfortable level of light for your eye quickly. And then the light source, if you need to make a more permanent change, there should be a dial to actually adjust the intensity that the bulb is putting out light. This slide is about how not to be the person who breaks a slide on the first day of class. So I've already mentioned, and it is important, that you only use the coarse focus, the big old handle to move the stage up and down, when you are using the 4x scanning objective. So if you are at a higher magnification, only use the fine focus. So as a matter of procedure, first you have to, once you put the slide onto the stage, you have to essentially just eyeball it from the outside. So move the stage very roughly until you see that the light is actually passing through the specimen. Then look through the eyepiece, center the specimen using the same controls, the XY stage controls. Then that's that. When you look through the lens, you can then focus using coarse, then fine, and if you need to go to a higher magnification, make sure that the object is very well centered, again, so that when you switch objectives to the higher power, it doesn't fall outside of the new field, because the new field is much smaller than the first field that you were looking at. Let's see, you should only have to change the focus slightly from there, because most microscopes are designed to have the same focal plane presented to the user. That's called being parfocal. And again, don't use the 100x lens. So these slides have their own explanations, so I won't really dwell on them as much, though I will try to abstract in another set of slides. But the point is, and I designed these to look like roughly the same change in magnification that you would see on our microscopes from 4x objective to 10x and 4x, 40x. So what we'll do when we're trying to estimate the size of an object is first understand how big the field that we're looking at is, and then compare an unknown object to that field. And the way that we do that is we go to the 4x scanning lens and we use a ruler. So when we lay the ruler over it, we have to measure at the very widest point of the to get the full diameter. If you measure up here, for example, well, I'm not using laser pointer. Wow. We'll see if that's important later. If you measure up here, you're obviously going to get a much shorter length than you would down here. So we want the full width, and you'll find that it comes out to some millimeters and some parts of a millimeter. So we'll record that, and then we'll actually calculate the size of the 10x and the 40x using the difference in magnification. So when you go, for example, from 10x to 40x, it'll be 4x, 4 times as magnified using the 40x lens. So objects will appear 4x bigger. However, the field will in fact be 4 times smaller. And that inverted relationship is what throws most of the students in this class. This, I'd say, despite this being heavily weighted on the test, only about 50% of the students get these points. So I want you to practice. I've created a separate exercise, and I will explain it to you as many times as necessary. So when we move up into 10x, that'll be about 2.5 times big over here. You see he looks bigger. And again, I would just have you trust these the text on the side for the actual calculations that you'll see in class. Note down to the 10x, you can actually still see a full millimeter on our ruler. However, a much bigger area of it would be uncertain, so it would be 1 millimeter plus, I don't know, is this a half, a third, a 4/5? I don't know. So it's actually much more accurate to calculate from the 4x back to the 10x. And with the 40x, you don't even have that option. So if we move to 40x, if a ruler were here, you wouldn't see anything. So you wouldn't see a full tick. So you know that you're just guessing if you wanted to say this was some number of millimeters. So we actually have to calculate back from the 4x for this example. So I really want to distill this for you. So there's only 2.5 jobs you have when you're trying to estimate the size of an object in your field of vision on the microscope. The first is to find a reference. The ruler is an example of that, but ultimately we'll use the width of the field of vision for that reference. The second job is to estimate what percentage of that field, or that reference, that your object covers. And then you simply have to multiply the two. So if your reference is 2 millimeters wide and your object takes up 1/2 of it, you know that it's 1 millimeter. So here's the problem in abstract. I ask you, how many units big is the blue ball? Hopefully you'll say that's preposterous. What kind of units? I have no reference, and so forth. So you need a reference. What if I tell you that that green circle is 2 units big? You would say, well, this blue ball looks apparently about 1/2 as big, and so if the green ball is 2 units big, then 1/2 of 2, there I am multiplying it, is 1 unit. And so part of it is a function of how good you are at estimating what percentage of that field is, but if you don't believe me that's 1/2, I'll just show you another one. Yes, it fits. So then here's the problem, where you're faced with estimating the size of an object, here the red object, compared to a known reference. So say we knew what the magenta circle was and the green circle was. separately. Which of the two references would you use to estimate the size of the red ball? I would hope that it would be easier for you to figure out that this is, say, 1/2 of the green circle versus one, I don't know, is it a 9th, is it an 18th? I can't do that. So I need a reference closer to the scale of the object for me to estimate appropriately. So here, then we can add the problem, well, what if we don't know how big one of the two objects is? So we can measure the size of the magenta object using a ruler like we will in lab, and we'll see that it's 1, 2, 3 units wide. And though we could kind of measure if we could see this whole green object here along with a ruler, we could measure the size of the green. However, that's not actually what's possible. So what this actually represents is this is the field of vision at a higher magnification, and this is the field of vision at a lower magnification. And no, I did not get that backwards, so if that doesn't make sense to you, stop and think about that. This is the field at a higher magnification. Objects look bigger because we're looking at a smaller field. This is what's represented by the lower magnification. You can fit more objects into it because you're not looking as closely. So the real choice is this. If you look at the magenta field, the low power field, you can actually see, well, it's some percentage of a unit measurement. But we know that the whole thing is 3. However, I'm still having a hard time estimating whether this is what percentage of the whole field or even of the whole unit. So it's actually much easier for me to appreciate that, yeah, yet again, this is about 1/2 the size of the total reference, which is the high magnification field. So this is where we enter the messy calculations. If we can't measure the size of the high power field, then we have to calculate it. So fortunately, we do have some other known entities, and that is the rate at which objects are magnified with those lenses. So the magnifying power of each lens is known. So the first low power is 4x, the second high power is 18x. And so if you divide these into each other, the 18x is 4.5 times as strong a magnification as the 4x, so objects will appear 4.5 times bigger. So then we can calculate if we know also the size of the field that we started with, the size of the calculated higher magnification field. And this is what it looks like. The low power is 3 units wide, and we know that we need to divide by 4.5 times. The field will be 4.5 times as small because objects appear 4.5 times bigger. That gives us 2/3 of a unit. Note that if we had a ruler here, we couldn't see it, a whole tick, which jives with the previous slide. And so then we do our second job, which is to say what percentage of the field is occupied, and that's 1/2. So we multiply those together. 50% times 2/3 of a unit is 1/3 of a unit is the total size of red, which actually it kind of jibes with what we see if we just eyeball it with a low power. So the other part of this chapter is to look at cells and tissues, and we're going to be looking at subcellular structures as well as the integration of whole tissues and the different kinds of cells that go into those. On this slide, I'll make a few points. First is mnemonics do matter. That's why I put this in here. You could actually remember that one of the stains, acetocarmine, if you can't remember just those words together, you might want to imagine what they look like. The second, and more substantial, is over here. I have highlighted the different cells that we look at and the features that are actually visible within those. So when we assess you on this, that is, on the test, when we show you onion epithelium, you can really only see 2 or 3 things, so you should be able to pick out those structures. And I've highlighted those in red. and I don't think that I should go into it in further detail. I will fly through images of those here. So Elodia cells, you can see chloroplasts, those are these things here. You can also see a large central vacuole, which we'll manipulate in the osmosis lab. In the onion cells, we're going to take the very thinnest layer of one of the shards of it, and you notice that with the acetocarmine stain, you can see the nucleus. And if you get in really close, you may be able to see the nucleoli. Red peppers we don't actually stain, but you can see the plastids, which actually hold the pigments that show the colors for. We'll stain potato cells with potassium iodide, and that reveals the starch capsules inside. You'll also want to note their shape when you get a cutting of these. Human cheek cells, will actually swab these off the side of our mouth, and about the only thing you can see is the nucleus and then the substructure, the nucleoli. Their shape is very important, and you should know what they were stained with, which is methylene blue. Yeast cells will be hard to look at. I believe we have prepared slides for these, but we will also look at electron microscopy images of them for their subcellular compartment. The yogurt bacteria we will try desperately to see, however, we probably will not because they are so small. But notice that they're linked together in rods here. Okay, so then finally there's two different tissues that we're going to look at. A gut epithelium, or rather a whole gut cross-section, as well as the cross-section of a leaf. And what you'll be responsible for knowing is what the different layers are called, what they are comprised of, what kinds of cells that is, and what kind of organization they have. So there are various tissue types, including epithelium, muscle cells, nervous cells, connective tissue, and others. See the full concept map for all that. But moreover, you're going to have to be able to identify those tissues from a microscope, not from a nice clean image here, with a little pointer pointing at a particular layer. You're going to have to describe the function, tissue type, and what it's called. So in order from the inside of the gut and going through their functions, there's an epithelium, which with a lot of surface area. You'll notice the wrinkles in this tissue, used to absorb nutrients. That's called the mucosa. Underneath that is the submucosa. This contains the vasculature to help take the nutrients away from the system. Also, it contains other connective tissues to attach it to the other layers. Moving backward, there are two layers of muscle. Those are called the muscularis, and those are arranged where there is a circular layer on the inside and a longitudinal layer, so running the length of the gut tube, on the outside. And I'm not going to go into it right now, we can discuss it, but stop and think. What happens when each of those muscles contracts in terms of the shape of the gut tube? And how does that get this single pulse pumping mechanism called peristaltic? How does it elicit that motion? But there are two layers of muscle there. They are smooth muscle, and it's called the muscularis again. On the outside there is a squamous epithelium called the serosa, and that is to protect the whole integrated organ/tissue. In our book there's one other layer called the lumen, and that's just the biological space inside of the gut tube. Again, you have to be able to look at the layers here and identify, based on the characteristics that you see, whether it's staining density or color or just their arrangement, density if I didn't say it, and so forth. The plant leaf, I think, is almost easier to explain from here. So starting at the top, there's a cuticle, which is secreted by an epithelium at the top called the epidermis. Underneath that is the parenchyma. It has different names for it. based on its density. So the mesophyll, the palisade mesophyll, is this stuff on top. It's called palisade. It's defensive. You can think of it that way. But principally what its purpose is trapping light inside the cell as it hits the light coming into it, or inside the tissue as it comes in. Beneath that is the much less dense mesophyll called the sponge mesophyll. That is not dense because its purpose is to maintain these air channels to let air diffuse into this tissue, so to let carbon dioxide in and oxygen out to allow photosynthesis to occur. You'll also note vascular bundles, and so the general rule is the living tissue is on the outside, that's the phloem. The dead or dying tissue inside is the xylem. Think of the tree when you cut it down. The living stuff is on the outside, underneath the bark, and the dead stuff is the wood inside. And the whole thing is wrapped by a vascular sheath. Note that there's different roles for what these things are transporting. The xylem on the inside is principally for water, and the phloem on the outside is principally for nutrients like sucrose and ions and such. Finally, there's a couple of structures to note on the bottom. The stoma are air holes comprised of guard cells, and again, that's for getting the carbon dioxide in, oxygen out. Here it is in cartoon. I think that's it for today. We'll check.